Hello and welcome to the fifth class of Immunology. In this class, we are going to discuss about immunosignaling. We will discuss what immunosignaling is, what are the components of immunosignaling, what are the different concepts of immunosignaling, and how immunosignaling can translate into an effector immune response. Okay, so now let us start with the previous class recap, what we discussed in our previous class, just to connect. So we discussed about the immunoglobulin superfamily. Okay, then we studied antibodies, their structure and their functions, their classes. We also studied about the B cell receptors and how B cell signaling activates a B cell, and then T cell receptors and their co-receptors and their signalings. Okay, so now we after discussing all the immunoglobulins, BCRs and TCRs, it is important to come to the stage where we will study more details about the cascade, signaling cascade, which takes place when any antigen binds to an immune cell, or when any foreign particle, or when any foreign body enters inside our body, and the whole array of our immune cells, they just pounce on that particular foreign body to eliminate it, to neutralize that, right? So this is important, this topic is important, because ultimately when we are talking about the immune response, then how immune response is generated, it is all because of the immunosignaling, right? It is all about the signal pathways. It is all about how antigen or a foreign substance is giving signals to our body to act upon it, right? So let us start with it. So we will see the aspects of signaling, and I think most of you have studied signaling pathways in molecular biology. So basically, molecular signaling is the term which we use for the signaling pathways, and immunosignaling is a very special kind of molecular signaling. So immunosignaling is the molecular signaling where we are concerned with the immunological responses, right? So it's a kind of molecular signaling only. So if we see the concepts or some basics of immunosignaling, then our cells, they sense environmental changes and respond suitably. Now, this is not only true for the mammalian cells, but also it is true for the small bacterial cells. They also have the system. And I'm sure you must be knowing about the two component system, where one component is the sensor protein and other component is the response regulator. So the sensor protein, it senses the environmental clues, the changes, and the response regulator, it acts upon it and it acts accordingly as per the signal. Now this is about the small word, this is about the very microscopic word we are talking about. Now we will see how the molecular signaling takes place in our body in terms of the immunological area. So now when cells this signal or they sense the environmental signal, they act accordingly, and this signal can be very very different. This signal can be related to the air, this signal can be related to some sort of food, this signal can be related to the environment or and the other environmental things. So it can be of anything. So when cells they sense the environmental signal, the signaling begins. And when signaling begins, only when there is a matching between the ligand and the receptor. Okay, the right ligand, when it binds to the receptor, then only the signaling cascade begins. Now if you see here, we have a signal. Now in this diagram, if you see here, so here is the signal. Okay, and this is signal is nothing but it is a kind of ligand, and when this ligand is binding to its receptor, so there is a receptor ligand binding, and when the receptor ligand binding is taking place, then only this interaction causes the signal into the interior of the cell. So this is the boundary. Okay, so you can say this is the cell membrane, This is the cell membrane, and this is the interior of the cell, though the outer signal binds to the receptor and then the internal changes takes place. Okay, and when there are so many relay molecules, so one molecule it activates another, another activates another, and finally they activate the effector molecule, which can be a transcription factor, and in most of the cases it is the transcription factor. And then this transcription factor, it goes inside the nucleus, and then it takes part in the transcription of a specific gene. So there are so many signaling molecules. For example, in case of nervous system, we see neurotransmitters. So you must have heard about the dopamine or acetylcholine or histamine. These all are the neurotransmitters. Then in endocrine system, we have the hormones as the signaling molecules. In immune system, we have antigens, which is a non-self entity. You remember that antigens are the non-self molecules, and this non-self entity, it is the signaling molecule of the immune system. And then we have the cytokines and chemokines, and actually the cytokines and the chemokines are the molecules which are the chemical messengers in case of our immune system. So they communicate among immune cells, and the chemokines, they are chemoattractant or chemorepulsive cytokines. Okay, so they both, cytokines and chemokines, they are the messengers, and they give the suitable and the appropriate message to the right cells, so that the right cells come into action, and then they start doing their job. So this is about some basics of the molecular signaling. Now let us see some concepts of molecular immunosignaling. So receptor expression impacts ligand interaction. Now this is very, very important. Receptor expression, any cell, if it will have an receptor, then only the ligand can bind. If the receptor is not there, then even the ligand is present in the environment, it is not going to bind and not going to impact the cell. So it is very important for the cell to express the ligand, right? So usually cells, they have a very low number of receptors which are expressed on the cells, and these receptors are not of much use unless the cell needs a particular desired product, and then in order to make that desired product through transcription, the cell increases the expression of the receptor, so that more receptors are increased on the cell membrane, and then the ligands which are there in the environment, they bind to the receptors, and then they start the signaling cascade. So receptor expression, it impacts the ligand interaction. Now some receptors are expressed only when needed. Okay, they are not at all present at all the time. So when they are needed, they will be expressed. If they are not needed, they will not be expressed. Now sometimes many signals through different receptors needed a particular outcome. It is not necessary that only a single ligand receptor interaction will give the outcome. There might be the possibilities, there might be some situations where many signals through different receptors, they are required when we need a particular outcome. For examples, we have TLRs and IFN gamma receptors on macrophages. Okay, now when these two receptors are expressed on the macrophages, then their combined effect is always greater than either of the receptors, individual receptors, is expressed. What does it mean? It simply means that when a macrophage, it has a TLR receptor, then also it will give some effect, okay, impact. It will have the IFN gamma receptor, then also it will give the impact. But the impact of both the receptors, when comes in combined, it will greatly enhanced. And the macrophages, they will be they will be very, very sensitive when they both the TLRs and the IFN gammas, both of the receptors are expressed. Okay, so when IFN gamma binds to the IFN receptor, that it substantially increases the TLRs, which are the toll like receptors, which we will be discussing in our subsequent classes. The toll like receptors are expressed, And when the TLRs are expressed, so TLRs are basically a kind of receptors which bind to the pathogen associated molecular patterns. And these pathogen associated molecular patterns, they are found exclusively on the microbes, right? And they are not expressed on the vertebrate cells. So when these receptors, TLRs, they determine and they bind to the suitable their PAMs or pathogen associated molecular patterns, then the signal transduction cascades begins, and then the macrophage becomes more sensitive and highly sensitive to give its effect to phagocytose the pathogen, as well as to produce the inflammatory reaction. Okay, so this example tells us that when the we need a particular outcome, and here the outcome required is that the inflammatory pathways should be enhanced, the inflammation should be greater, and the microbe should be eliminated. For that particular purpose, both the receptors, they work in sync. Okay, now, the next is a lymphocyte has a unique B cell receptor and T cell receptor. So, B cells will have a unique BCR and the T cells will have a unique TCR, but many receptors for cytokines. Okay, on a B cell, there are BCRs which are specific for only one antigen. On the T cells, there are also TCRs which are specific for only one antigen. But there are so many other receptors which are for different cytokines and for different chemokines. Now, what happens that the cell must integrate, okay, the signals from all the receptors occupied at once. So this is the part which the cell plays to take a right decision, that when it has so many ligands which are attached to the receptors, then cell integrates all the signals and then takes the appropriate decision that on which path it has to go, whether the path is to increase the immune response or the path is to decrease the immune response. It will all depend, what kind of combinations are taking place at that particular point of time and what kind of ligands are present in the environment. Okay, now growth factor receptors, they have inbuilt inducible enzyme activities. So most of the growth factor receptors, GFRs, they have inbuilt inducible enzyme activities, and these enzyme activities, they act upon the different molecules, and then we call them different adapter molecules or the associated molecules, and there is a cascade of phosphorylation or dephosphorylation takes place, and then the particular cascade or the particular path is taken. Then on BCRs and TCRs, they have the short cytoplasmic tail, and the signal transduction is by receptor associated molecules. There are so many receptor associated molecules which are found along with the BCRs and TCRs, and then they are involved in the complete signal transduction pathway. Now, if you can see here on B cells, there is Ig alpha, Ig beta, which we call as CD79 alpha beta heterodimer. So you can find this Ig alpha and Ig beta here. Okay, now these are the molecules which are involved. Okay, and in case of the T cells, there is a hexameric CD3 complex. So you can see here, this is the CD3 complex, and this CD3 complex is hexameric. Why? Because hexa means 6. So 1 2 3 4 5 6, it has 6 units. So these are required, and they are required because they bind with so many different molecules, which we will see when we will see the signal pathways, and then with the help of those molecules, the whole signal transduction takes place. Okay, now receptors bind to ligands using non-covalent bonds. Okay, receptors, they bind to ligands using non-covalent bonds. So here you can see this is the receptor and this is the ligand, and this ligand is binding to the receptor. Now what you can see here is two things. Number one, that receptor is binding with the use of the non-covalent bonds, and these non-covalent bonds or non-covalent interactions, they are very weak interactions. I'm sure you must have, studied this thing, that non-covalent bonds are the weaker bonds as compared to the covalent bonds. But their strength lies in the number of interactions. That means more number of non-covalent bonds make a stronger connection, make a stronger interaction. So number of interactions is proportional to the strength. More number of interactions, more strength. Less number of interactions, less strength. Simple. And then complementarity is the key. Complementarity is what? That the shape of this particular ligand, it complements the shape of this receptor so that it can easily fit into it. And when it will easily fit into it, and it will have, the perfect complementarity, then only the interactions will increase. So the number of interactions will only increase when there will be the complementarity. And when there is a complementarity, then only the receptor can bind to its ligand. Okay. Now here you can see the a number of non-covalent bonds which are involved in the receptor ligand binding. There are hydrogen bonds, then we have ionic bonds, then we have hydrophobic interactions, we have van der Waals interactions, and then we have the ionic bonds. And they all contribute to the receptor ligand binding, right? Successful receptor ligand interaction means the interaction holding both with enough binding energies. When we say that the binding or the interaction is successful, it means that there are enough bonds which can hold the, the receptor ligand complex in a stable manner. And how it can happen? It can happen by the number of interactions and the complementarity. Okay, that is the key to any successful ligand receptor interaction. For sufficient time to allow the signal to transduce. Sometimes what happens, that there is a binding, but this binding is not for sufficient time. It is binding good, but then it is immediately dissociating, then binding, then dissociating. So this dissociation and binding, it is not going to help. Why? Because it needs some time so that the signal, when it comes in its appropriate form, and how it comes to its appropriate form? By the changes in the conformation of the protein or the receptor. So it will only take place when there is a sufficient time given to the receptor, when the ligand binds to it, and then only the conformational changes take place, and then the downstream cascade begins. Okay, so for sufficient time to allow the signal to transduce. So these two are the conditions, the interactions holding both with enough binding energies and for sufficient time. When these two conditions are met, then the likelihood of the likelihood of signal transduction successfully taking place will increase, right? Now, let us see, if R is the receptor and L is the ligand, then the binding equation is R plus L is equal to RL. Okay. Now, here is the K1, which is the forward or association rate constant, and K minus 1 is the reverse or the dissociation rate constant. That means this is a reversible reaction. It can go in forward direction also, and it can go in reverse direction also. The ratio of K1 to K minus 1 is known as the association constant of the reaction. And this association constant, it measures the affinity of the receptor ligand pair. Okay. Now, Ka is a relationship between the concentration of the reaction product, which is RL. So here you can see this is your RL, and the product of the concentration of the reactants R and L. So here it is RL divided by R multiplied by L. The reciprocal of the association constant is the dissociation constant Kd. So Kd is inversely proportional to Ka. The higher the Ka or lower the Kd, the higher the affinity. Clear? So when we have the higher Ka, the affinity is higher. Or when we have the lower Kd, the affinity is higher. In both the cases, the affinity is higher or vice versa. Right? Now, let us understand the difference between the affinity and avidity. So, affinity is the strength of the binding between a single binding site on a molecule. So, suppose this is an antibody molecule, right? On this antibody molecule, we have two binding site. This is binding site number 1 and this is binding site number 2. So, what is affinity? Affinity is the strength of the binding between a single binding site on a molecule. So, on a single molecule having two binding sites, affinity is the strength of the binding between the single binding site on a molecule. Now, it measures the individual interaction. Okay, it is described by the dissociation constant Kd. So as we just discussed, lower the Kd, higher the affinity. Okay, now what is avidity? Avidity describes the overall strength of the collective binding interactions. Okay, here the important is collective binding interactions that occur during multivalent binding. So multivalent binding is what? When on a molecule more than one binding sites are occupied by the ligand. So in this case, this is binding site number 1. Suppose this is, here we have a antigen. So here also we have an antigen. So when we see their combined binding, then we see the term we use is the avidity. But when we use or when we just are concerned with the binding site which is just single binding site on a molecule, then we use the term affinity. Okay, sorry, when we use the term affinity. So avidity is the combined strength of the multiple affinities. This is important. The combined strength of the multiple affinities is the avidity, and higher avidity can occur even if individual affinities are low. Why? The same thing what we discussed. The non-covalent interactions, although they are very weak, right, but still when they have a higher number of interactions, then it gives them the stability. Okay, they are stronger, much stronger. In the same manner, here if we see, the individual affinity may be weaker, it may have a very less strength, but when so many affinities are seen in toto, then the complete avidity, when we assess, then it will be much higher. So higher avidity can occur even if the individual affinities are low. It is also referred to as the functional affinity, right? Avidity is also referred to as functional affinity. Other co-receptor interactions can also contribute to the overall avidity of the engagement between a cell and its antigen. So it is not only the main receptor. For example, it is not only the BCR or the TCR, but then there are so many co-receptors which are always there. For example, in case of T cells, it is the TCR and then we have the M.Sc molecules, right? And these M.Sc molecules and TCRs, they together, they make a very good strong interaction between the antigen and their respective receptors. So co-receptors, they also contribute to the overall avidity of the engagement between a cell and its antigen, right? Now, many biological receptors, for example BCRs, they are multivalent. When we say multivalent, what does it mean? That means they have more than one ligand binding site per molecule. More than one ligand binding site per molecule. Okay, for example, we saw that in antibody there were two binding sites for the antigen, right? So it is a multivalent, we call it as a multivalent interaction. So here you can see these are the binding sites and this is the ligand, okay, and where it is binding. So this molecule has the two binding sites, right? The overall binding interaction is significantly enhanced compared with a similar but univalent interaction. Okay, so here you can see this is the single binding site. When it is binding, the situation is in on condition, but when it is dissociating itself, then it is in the off situation. So it is either on or either off, but here the case is different. Here, even if the binding site is half of the binding is occupied, that means out of two, one is occupied, still it can give the signals. But when both are occupied, then it is a greater signaling. Okay, so cell membrane is fluid. Cell membrane is fluid. So antigen receptors act in a multivalent manner even if monovalent. Right? This is important because we know that our cell membrane, it is in fluidic, it is of fluidic nature. So when antigen receptors, they are monovalent, but still they behave like a multivalent. How? You can see here. This is the antigen and it is binding to the antibody molecule. But when it is binding, then actually there is a clustering of the antibody receptors happening here. So this is BCR, and here you can see the BCR clustering is happening even if there is a single antigen which is binding to the antibody or the BCR. Sorry, I should use the term BCR here because this is the membrane bound antibody molecule. So you can see here the single antigen, it is causing the clustering of the BCRs, and then it is making the interaction as a multivalent interaction, not as a univalent interaction. Okay. There are unlimited antigens or diverse signals, but we have finite receptors. Right? We don't know how many antigens are there. There may be millions of antigens, right? There may be millions of different foreign particles which can enter inside our body, but our body cannot make the receptors for each and every antigen. So our receptors are finite, they are limited in numbers, but the antigens or signals are unlimited in numbers. So how our body deals with it? So there is a combination of one receptor chain with different partner chains to increase the receptor diversity and affinity. That's a very smart way. What is happening here, that we are combining one receptor chain with different partner chains so that we can increase the receptor diversity and affinity. So in this manner, although we have the limited number of receptors, but still we can increase our diversity. Now let us see how. So you can see here we have a low affinity receptors. This is the alpha chain, this is alpha chain, this is alpha chain, and this is for IL-3, this is for IL-5, and this is for GM-CSF. Okay, but when we combined it with the same beta subunit, so beta subunit is the same, this is beta, 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 then that they change from low affinity receptors to high affinity receptors. So now this alpha beta combination is giving more affinity than the individual low affinity receptors. So we what is happening here is that the partner chain, this partner chain, when it is coming, it is the same chain here, beta, beta, beta in all these three cases. So the single chain, it has increased the affinity rather than giving the individual chains to different receptors. Our immune system avoids response to an irrelevant antigen. Now this is a mechanism which is very important for our body. It avoids response to an irrelevant antigen, the antigen which is not required, or the antigen which is not, which does not need any immune response. For example, IL-2 signals, IL-2 is our cytokine. It signals our B or T cells to initiate proliferation and differentiation. Now, naive cells are those cells which are unexposed to the antigen yet. Those are known as naive cells. So naive cells, they express a heterodimeric intermediate affinity form. Heterodimeric means the dimer has two different chains, right? Then intermediate affinity, neither low affinity nor higher affinity, in between, intermediate affinity form of the IL-2 receptor, and this is known as IL-2R. This is IL-2 is the name of the cytokine, R is receptor, and two chains are beta and gamma. So that's how they are named. They are named. So this is IL-2 R beta gamma. So there is no response to physiological concentration of IL-2. Okay, when there is an intermediate receptors, then there is no response, even if it is binding, but the response will not come here. So you can see here, this is IL-2R, this is IL-2R, this is IL-2R, but this is the low affinity because it has a single chain, right? Then when the intermediate affinity, it has IL-2R beta gamma. Here it is beta chain, this is gamma chain, and this is of the intermediate affinity. So we are talking about this particular thing, intermediate affinity. Now when antigen binds, okay, it activates Bt cells and combine a third chain, which is IL-2R alpha. So you can see here, we already have the gamma chain and beta chain, gamma chain and beta chain here in intermediate. But what additional chain is expressed is now the alpha chain. So this alpha chain, it combines and it converts the intermediate affinity to high affinity receptor. Okay, so antigen activated B or T cells, they combine a third chain IL-2R alpha with the other two chains, converting into high affinity form, which can now respond to IL-2. Okay, now why it is important? Because it conserves the energy and then no accidental initiation of an immune response. Otherwise, we will have our own cells or own tissues damaged, right? So this will be a different condition, which is a immune dysfunction, if this kind of things happen, or misfunction. But it makes sure that no accidental initiation of immune response happens here, as well as our energy is conserved. All right, very smart way. Now let us also see some properties of immunoreceptors. Our immune receptors may be located on the plasma membrane or on intracellular membrane, in cytosol, or they may be floating in the tissue fluid. They may be anywhere. All the immune receptors, they may be anywhere. Okay, so here you can see we have the intracellular receptors, we also have the receptors on the plasma membrane, right? And then we have the receptors here also, so it can be anywhere. Now we have two kind of receptors, one are innate receptors and other are the adaptive immunity receptors. So let us just compare the difference, or the comparison between the innate and the adaptive immunity receptors. Innate receptors are expressed in non-clonal manner, right? You understand clonal expansion? We studied in the first class what was the clonal expansion. Okay, so they innate receptors, they expressed in a non-clonal manner. That means they are expressed in all sort of cells and they are not clonally selected. While in case of the adaptive immunity, they are clonally expressed. So we have one cell, one receptor, and this one receptor is BCR or TCR, BCR or TCR, BCR on B cell and TCR on T cell. And when one antigen, it binds with either BCR or with the TCR, then the cell is clonally selected, and then it is made into the cell division, which makes the identical cells, which will have the same specificity of the receptors. Now, innate receptors, they are expressed on diverse immune and non-immune cells, and we have more than around 100 cells as of now, which are discovered so far, right? While the B cell receptors and T cell receptors, they are bound only by the B lymphocytes and the T lymphocytes, but they are the billions of cells. Okay, and when we have the billions of cells and these have the BCRs and TCRs, so the diversity here is much more. Okay, we have the PRRs. So PRRs are the pattern recognition receptors. So pattern recognition receptors are the receptors which are present on the innate cells as well as on the adaptive immune cells, and they recognize PAMs. PAMs are the pathogen associated molecular patterns. PAMs are pathogen associated molecular patterns. There are certain conserved patterns which are present on the microbes which are not expressed on vertebrates. Therefore, these patterns or these exclusive entities are easily identified by our immune cells using the receptors which we call them as PRRs, which are the pattern recognition receptors. And these pattern recognition receptors, they recognize PAMs, which are pathogen associated molecular patterns. In general, we call them as MAMs, which are the microbe associated molecular patterns. Why? Because not all microbes are pathogen, right? Not all microbes are pathogen. So when we are using the term either PAM or MAM, they will both, we can use both the terms. But when we know that a certain microbe is a pathogen, when we use the term specifically as PAM, but if we don't know, or if it is not at all the pathogenic but still it can bind and give some response, then we call it as a MAM. So PAMP is pathogen associated molecular pattern and MAMP is microbe associated molecular pattern. Or PRR can also recognize DAMs. What are DAMs? These are the damage associated molecular patterns. DAMP. DAMs are the signals from damaged cells, dying cells, or dead host cells. All right. So these are damage or danger associated. Sometimes we also call them as a danger associated molecular patterns or damage associated molecular patterns. And they are usually they are found at the time when there is a immunological fighting going on and the tissue is also injured. So these are required for healing of the tissue, right? And for the clearance of the dead cells or the dying cells or the dead immune cells or the dead pathogens, right? So these are the PRRs. PRRs are present on our immune cells. They are again I'm telling you, they are the pattern recognition receptors, and they recognize the PAMs or MAMs or TAMs. All right. Some PRRs are expressed on internal cell membrane, which are like endosomes, or in the cytosol, which are capable of binding viral RNA that might never be expressed on the membranes. Because you know that the viruses are a very unique cases. They go, they attack the host cell, they go inside the cell, they just take over the machinery, and then they start their synthesizing their own nucleic acids, and these nucleic acids are never expressed on the membrane. They remain inside the cell only, but these can also be bound with some PRRs which are there. So our immune system have that capability that our PRRs are expressed not only on the outer membrane of the cells but also inside the cells, where they can easily bind with these viral nucleic acids like RNA. And once they bind with the RNA, they start providing, or they start the appropriate immune response. Now, if you see the adaptive immune receptors, we have the BCRs and TCRs, and these BCRs, they recognize epitope on an antigen, while TCRs recognize MSC presented peptides on the surface. So I think we have just in our last class we discussed these two things in a bit details. Okay, so this is the difference between the innate receptors and adaptive immunity receptors. Now let us just focus on the signal transduction pathway. So what is a signal transduction pathway? It is a molecular route, okay, by which a ligand receptor interaction is translated into a biochemical and behavioral changes within the affected cells. Very beautiful definition. What is happening here? Signal transduction, it is a pathway. So when we are using pathway, that means it is a route. So it is a molecular route by which a ligand receptor interaction, which is happening outside the cell, on the membrane, it is translated into a biochemical and behavioral changes within the affected cells. So we have the upstream components which are closest to the receptors, and then we have the downstream components which are closest to the effector molecules that determine the outcome of the pathway. So ligand receptor binding, it induces a conformational change in the receptor, mostly causing receptor polymerization. So receptors are usually not polymerized unless and until the ligand binds to them. And when they binds to them, then it causes their polymerization, which leads to even the univalent interaction into a multivalent interaction, which we just saw a couple of slides back. Okay. Now here you can see this is the BCR with the associated molecule, and they are just the individual receptors. But when the antigen is binding, then what is happening? There is a clustering happening, or the polymerization of the receptors taking place. Okay. Now, we have lipid rafts on the membrane, and lipid rafts are very important for the signaling. How? First of all, let us understand what lipid rafts are. So, lipid rafts, these are the regions which are rich in saturated lipids, cholesterol, and glycosphingolipids. So here you can see we have the membrane. In this membrane, this particular region is the raft region, from here to here. Now in this region, if you see, you can see here the cholesterol, this blue color entities. So lot of blue color entities are there within this region. Then we have saturated lipids, and this is the sky blue color. You can see here the light blue color, saturated lipids a lot here than the neighboring region. Then we have the sphingomyelin. All right, so you can see here a lot of sphingomyelin here within this region, right? Then we have the gangliosides. You can see some gangliosides here within this region, right? Then we have the transmembrane proteins and the channels and some other proteins which are present and which are rich within this region. So this is called as a raft region. And when you see the neighboring region, so the neighboring region is not this much rich. Okay, so first of all, you understand what a lipid raft is. So this is the lipid raft. Now this raft is populated by signaling molecules. Populated by signaling molecules. That means it has a lot of signaling molecules which are restricted. I mean, which are, I would not say the restricted, rather than they are, the likelihood of having these signaling molecules are much higher here, and they are present here in higher concentrations. For example, tyrosine kinase, which is lin. So lin is a kind of tyrosine kinase enzyme. In resting B or T lymphocytes, BCRs and TCRs are excluded from the rafts. Important. In the resting condition, okay, when the B or T cells are not interacting with any antigen, then the BCRs, that is the B cell receptor and T cell receptors, they are excluded from the rafts. And when there is an antigen binding, it causes the BCRs and TCRs to cluster and increases their affinity to the rafts. So you can see here, this is the BCR, okay, just an example, and this pink region is the raft region where you can see we have the lin. So here the antigen is not bound yet. So they are outside the raft region. But when the antigen is bound to the BCRs, then they have more affinity towards the raft, and then they are attracted towards the raft. Now BCRs and TCRs, they move into the raft and contact with molecules like tyrosine kinase lin, which phosphorylates the receptor associated proteins Ig alpha, Ig beta. So you can see here this lin is here, which is present in the raft. As soon as they are coming inside the raft, this lin region, this lin enzyme, it phosphorylates the associated proteins Ig alpha, Ig beta, okay, and then it initiates the activation cascade. So the lipid rafts, they are very important for signal transduction process. They have a very important role here, right? Now, there are few signal transduction pathways which are repeated in cellular responses to different ligands. Okay, so there are different ligands, but the signal pathways, they are repeated for the in the cellular responses. These are utilized in different forms in multiple cellular systems and in many species. Okay, understand again. There are few signal transduction pathways which are repeated in the cellular responses to different ligands. So ligands may be different, but the signal transduction remains the same. They are repeated and they are utilized in different forms in multiple cellular systems and in many species. Okay, now immune pathways, they are triggered by antigen binding to the receptor and leads to the activation of different family of transcription factors. They are triggered by antigen binding to the receptor and it leads to the activation of different family of transcription factors. So these transcription factors, they upregulate or increase a cascade of important immune response genes, and the framework of these pathways is shared among many cell types with small modifications and cell type specific transcription. So more or less these signal transduction pathways, they remain the same, but there are few changes or few modifications, based on the kind of cells and the kind of different specific transcription process. Clear? Now, we have three pathways of particular relevance to the adaptive immune system, where we see a very significant importance here. First is the PLC pathway, second is the RAS MAP kinase pathway, and the third is the NF kappa B pathway. So we will see all these three pathways and how these are important, how they are translating into the immune effector mechanisms. Okay. Now, let us understand the general PLC pathway, not the immune specific, but what is the general PLC pathway? We have the cell membrane phospholipids, and phospholipids are present in all the cell membranes, right? Their cell membranes are rich in phospholipids. There are structural variations in hydrophilic head. So phospholipids, there are different kind of phospholipids and the structural variation in their hydrophilic head. For example, phosphatidyl inositol (PI) or phosphatidyl choline (PC). These are the two examples of the phospholipids. The structural variability facilitates their functions, including cell signaling. Okay, so when we need, or different type of their heads, they can have the different outcomes because they actually impacts or they influences what kind of signal pathway will take place. Phospholipases are the enzymes, so we call them as PLs. Phospholipases, they hydrolyze phospholipids by cleaving the ester bonds. We know that the phospholipids have the ester bonds, and phospholipases are the enzymes which they hydrolyze the phospholipids. And how they do? They do by cleaving the ester bonds. Now, based on different sites of cleavage, the phospholipases are classified as phospholipase A, B, C, and D. Okay, so PLA, PLB, PLC, and PLT. They generate secondary messengers for cell signaling, membrane trafficking, cell proliferation, etc. The role of these phospholipases is to cleave the phospholipids and then generate the secondary messengers, and these secondary messengers are different secondary messengers based on what kind of phospholipases are involved, whether it is A, B, C, or D. And then these secondary messengers, they can have different impact. They can They can be utilized for trafficking, for cell proliferation, for cell differentiation, and so on. Okay. Now we are concerned with the PLC, right? So PLCs, they hydrolyze PIP2, which is the phosphatidyl inositol 4,5 bisphosphate, to generate two secondary messengers. One is DAG, which is diacylglycerol, and another is IP3, which is inositol 1,4,5 trisphosphate. Okay, so this PLC, it hydrolyzes PIP2, and then it generates the two secondary messengers. One is DAG and another is IP3. So we are concerned for our immune discussion about this particular phospholipase. So that's why we will just discuss the PLC pathway. Now let us understand the PLC pathway in T cells. Now there is a T cell stimulation, right, by binding. Now tyrosine phosphorylation of the ITAM of CD3. This is ITAM of CD3. It causes the localization of the adapter protein LAT to the membrane. So you can see here, this is the phosphorylation and the LAT has come here to the membrane. Now in turn, LAT is phosphorylated, so this LAT is also phosphorylated, and PCL1 binds to it, having access to its substrate PIP2. So here it is binding after phosphorylation. This is binding, and now it has the access to the substrate, which is PIP2. So here is the substrate PIP2. Now, PCL1 is activated by tyrosine phosphorylation by kinases LCK and ITK. So here you can see this is the LCK and here it is the ITK. So they too are important for the activation. Alright. Now activated PLC1 mediates cleavage of PIP2 to IP3 and DAG, which are the secondary messengers. So here you can see the PIP2, it is degraded, or it is cleaved into two secondary messengers. One is DAG and another is IP3. Clear? Now calcium ion released from endoplasmic reticulum. We have endoplasmic reticulum here and it has released these calcium ions. They bind to the signaling intermediate calmodulin. So there is a signaling intermediate and we have this calmodulin which is inactive, and this calmodulin is binding with this calcium ions. Okay, and the calcium ions just want to make, because this pathway needs the activation of the calmodulin, so we need this calcium ions. Now the calcium calmodulin complex binds and activates the phosphatases calcineurin. So here you can see this is the calcineurin, which is the phosphatase. So it is actually, first it is binding, calcium is binding to the inactive calmodulin, right? And then after binding, this bound and activated calmodulin, it is also activating the calcineurin, which is the phosphatase, and this dephosphorylates the transcription factor NF80. So here you can see the NF80, it is already phosphorylated, but it dephosphorylated, that means it is taking out the phosphate from the NF80, and then that is the main function of this calcineurin. Now the dephosphorylated NF80, it enters the nucleus. So you can see here this NF80 is entering into the nucleus and activate the transcription of the important T cell target genes like IL-2. So because this is the transcription factor, so it is going binding to the DNA and then it is activating the transcription of the IL-2. Okay. Now, let us study the second one, which is the RAS MAP kinase pathway. Now, RAS is a monomeric GTP binding protein. We call them as a G protein. And the GDP bound form is inactive and the GTP bound form is active, right? If it is bound with GDP, then it is inactive. If it is bound with GTP, it is active. So here you can see this is the inactive form because GDP is bound, and here it is the active form because the GTP is bound. All right. Now, G proteins, they have a core GTPase activity further improved by GTPase activating proteins. We call them as the gaps. So these gaps, they improve the core GTPase activity. Now gaps inhibit the protein's activity by stimulating RAS intrinsic ability to hydrolyze bound GTP. So what is happening? It is actually, this gap, GTPase activating protein, it is inhibiting, okay, it is stimulating RAS intrinsic ability to hydrolyze bound GTP, and basically it is making it inactive. Now we also have another, which is guanine nucleotide exchange factors, which are GEFs, which act in opposite manner to release the GTP and promote GTP binding. Okay, so now we have the GEFs also, and they promote the GTP binding. So GTP, kicks away this GTP and bound to the RAS, and then it activates that. Okay, so it activates RAS by inducing it to release the GTP and accept GTP. So this is how it is happening. The RAS is coming into the active and inactive form with the help of the gaps and the GEFs. All right. This pathway is initiated during both B and T lymphocyte activation. Right. Now, see this pathway in details here. In T cells, DAG, which was released in PLC pathway, it binds and activates RAS-GRP, which is an adapter protein that recruits the GEF, which is called as SOS. So here you can see this is the RAS-GAP and this is the SOS. So they are recruited here, and this is the DAG. Right now, this SOS, it binds to RAS, inducing it to bind to GTP. So now this is basically inducing it to bind to GTP, and RAS activates the RAF. And what is RAF? RAF is map K K K. So you can see here MAP, then 3 times K, 1 2 3. So we call it as a map kinase kinase kinase. So 1K represents 1 kinase. So there are 3 Ks, so there are 3 kinase. So map kinase kinase kinase, or map KKK. Okay, this is the first member of a cascade of serine threonine kinase enzymes that phosphorylate and activate one another. Okay, so what will happen? It will again, this is a kind of serine threonine kinase enzyme. It will phosphorylate and activate another one. So now RAF, this is RAF, it phosphorylates and activates the MEK, which is a map kinase kinase. This is map 2 times K, KK. So here it is 3 times K. Now it activates 2 times K, KK, MEK. All right. Now, the activated MEK phosphorylates ERK, which is a MAP kinase, which can enter the nucleus. So this is now ERK, and it has just 1K. It is MAP kinase, and this ERK, it can enter inside the nucleus. In nucleus, ERK, it phosphorylates and activates a transcription factor, which is ELK1, which cooperates with the second protein SRF to transcribe a phos gene. So here it is ELK1. Okay, now this is phosphorylated, and when it is phosphorylated, it cooperates with the second protein, which is SRF. Okay, and these two together, they actually transcribe the phos gene to make a phos product. Now phos is phosphorylated by ERK. Here we have the phos, and it is phosphorylated by this ERK again. So you can see the phosphorylated phos with its partner Jun. Okay, here it is the Jun. It forms the master transcription factor AP1. So this is AP1, and this AP1, it has Jun plus phos in both in the phosphorylated state. Clear? Now, AP1 is one of the transcription factors that facilitates the transcription of the IL-2 gene. So this is basically, IL-2 gene. So now the IL-2 cytokine is produced here. Okay, so this is the RAS MAP kinase pathway. Now, the third pathway is NFKB pathway. So, pathway is activated when DAG generated by PLC, it recruits the serine threonine kinase PKC theta to the membrane. So, you can see here PKC. All right. Now, activated PKC phosphorylates adapter protein, which is karma 1. So, you can see here, this is karma 1. It is phosphorylated. So, here you can see the phosphate group. It is phosphorylated and initiating a cascade that at last recruits the ubiquitin ligase TRAF6. So, after a number of different proteins, ultimately it is the TRAF6, right? Now, TRAF6 ubiquitinates and partially activates the IKB kinase, which is in the IKK complex. It has a regulatory protein NEMO. So, you can see here in this complex NEMO and two catalytic IKK subunits. So, this is IKK alpha and IKK beta. All right. And these three make the IKK complex. All right. Now, other signals, they activate the TAK1 complex. Now, this is TAK1. This is TAK1 complex and it is activated by other signals and it phosphorylates the IKK. So, you can see here IKK is phosphorylated. Phosphate groups you can see here. This IKK complex is fully activated now and as it is both phosphorylated and ubiquitinated. So that means in order to activate the IKK complex, it has to be phosphorylated and it also has to be ubiquitinated, right? When these two conditions are met, then the IKK complex is activated, fully activated. Okay. Now this IKK phosphorylates and inactivates IKB, allowing translocation of NFKB into the nucleus. So you can see here, this is NFKB, it is now entering into the nucleus. Now this in nucleus signals transcription of its target genes, including the IL-2 genes. So IL-2 genes are again, the target of this particular transcription factor. Okay, so this is the third pathway. Now you can see the integration of these pathways. So you can see here the DAG, here it is the PKC, this is the RAS, and then this is where the NFKB, this is AP1 and NF80. So this slide basically summarizes how in a lymphocyte the signaling pathways they take place. We studied the individual pathways and now we see the all three pathways in a single slide. So ultimately what is happening? Ligand binding is happening here, okay, and then there are relays of molecules and ultimately what is happening is the gene activation in all these three pathways. If you can see here, gene activation, because these are the different transcription factors and these transcription factors, they are doing the transcription or activating the transcription of the different genes which are required. So this is how the signal transduction pathways, they translate a particular signal or ligand receptor interaction into the specific immune response. Okay, now so just we close our class today here and let us summarize what we have learned so far. So we discussed the molecular signaling, then some important concepts of immunosignaling, then receptor ligand interactions and some concepts about receptor ligand interactions, then fundamentals of signal transduction, and then we studied the three pathways, the PLC pathway, the RAS map kinase pathway, and NFKB pathway. Okay. I hope you have had some idea now about the signal transduction. So in case if you have some feedback about the class, you can always reach out to me in the given email address and let us meet in the next class. Okay, till then, take care and thank you.